So moving forward, Golia Shafiei sadly cannot be with us, but luckily Andrea agreed with her that he will take her talk on her behalf. So I will introduce Golia and then Andrea will take it away. Golia Shafiei is a postdoctoral fellow at the University of Pennsylvania, and she's working in the lab of Dr. Ted Satterwhite. And her research focuses on the intersection of multimodal neural dynamics, brain development, computational psychiatry, and large-scale data science using multiple neuroimaging modalities. So Golia, Andrea, please.
All right, yep, turns out you're stuck with me again. Hopefully I will do justice to Golia's work. It really is wonderful work, so I'll try to represent it as best I can. Now, Dr. Shafiei and I have been working together for a few years now, and something that is of interest to both of us is the question of regional heterogeneity across the brain. Because we know that the brain is not uniform. Different regions have different local properties, and not just across one dimension, but across many of those dimensions. And although they can all be summarized across large-scale axes, for example, this very famous sensory to association axis, nonetheless, they each are different, and they each have different meanings. And the question, therefore, arises, well, what can we do with them? What do they tell us about the way that the brain works? We have molecular and cellular properties, and those properties shape the circuit properties. So we're gradually abstracting away. And finally, and inevitably, they will shape the recordings that we get from brain activity, regardless of what modality that is, but perhaps in ways that are not identical to each other according to modality. But we also know that those local properties do not live in isolation. Rather, as we've been discussing multiple times already this morning, the brain is a network, and it can be recapitulated in terms of many of these properties relating, despite being local properties, relating to the local and global network embedding in the brain, not just in human, but also in macaque or mouse, for example, and every other known species that we can think of. And in fact, we don't just have structural networks, we also have functional networks. And those local microstructural and local annotations also relate to the functional networks of the brain. And that can be imbued in local models to try and understand what those properties are giving us in terms of computation and arguably in terms of overall brain organization. So altogether, what this talk is going to be addressing is what is the organization in terms of topography of the brain's intrinsic dynamics across the cortex? How are those cortical microarchitectural properties, how do they signal their own presence in terms of neurophysiology? And what can we do with them for understanding perturbations of the brain, which once again is the topic of this symposium. So let's get started in terms of reconsidering our relationship with time series. Time series is what we acquire from the brain to look at how it acts. We have many modalities for them, and we have many ways of looking at them and analyzing them. For example, functional connectivity, the cross correlations between them, how similar they look to each other over time. But we can also look at local time series properties, and especially in MEG and EEG research, it is very common to divide them in frequency bands and look at their spectral properties. But those are not the only ones that we can deal with. Because when you're thinking of time series, there's actually a lot that can happen in time. For example, just like we have visual receptive windows in the visual system, we also have temporal receptive windows that mirror the spatial dimension, but in time across the cortical hierarchies. Once again, we can look at the power spectrum characteristics, but we can also look at a whole lot of different time series properties, whether that's the distribution of the data, whether that's the relationship between the past and the future, the complexity. So rather than just hand-picking measures, another way that we can approach, perhaps a more principled way, is, for example, highly comparative time series analysis, a method developed nowhere else than here in Australia. And we can ask, what are those other properties that we've not been looking at? And it's not just one or two properties that we might have been neglecting, but actually you can systematically phenotype the whole dynamical landscape of a single time series into more than 7,000 dynamical features, which is a lot of features. And you can then use those features to compare different brain regions or different phenotypes in terms of their systematic signature of dynamics. And so this is the approach that Dr. Shafiei took already starting 5 years ago in this eLife paper. And the question was simply, what is the overall cortical organization of those features? Are there systematic patterns, or are they instead just distributed at random? And you can use PCA to provide a lower dimensional representation of their topographic organization. And you find that there are two significant components, one primarily accounting for different types of autocorrelation, and another one accounting for different data distribution properties. And you can then ask, how do those individual principal components, large-scale representations of the topography of dynamics, so we're blending space and time, how do they relate to those underlying biological properties of the brain that explain why the brain is heterogeneous? And in fact, we find that both of them have significant relationships with underlying representations of brain architecture. We have genetic representations, but we also have microarchitectural representations, including the brain's unimodal, transmodal hierarchy, for example, obtained from myelination, but also from function. So altogether, we can see that we have dynamical fingerprints of different regions that are different across the cortex, but not at random. Rather, they are systematically organized across the cortex in a way that recapitulates structural and functional properties of local regional heterogeneity. But all of this was based on functional MRI data. What about other modalities? I've been saying that we have different ways of looking at brain dynamics from different modalities of acquisition. So let's look at MEG, which is a lot faster and potentially richer representation of the activity of the data. It's also not hemodynamic, but it's a more direct representation of how brain regions act and interact. So we can take the same approach, hemodynamic responses, MEG responses to the activity, and we can systematically phenotype those time series activity for every region with thousands and thousands of different dynamical features. And I've been railing before against the use of hand-picked measures of characterizing dynamics, but then again, I've been talking to you about the use of hand-picked measures for characterizing brain microstructure. Well, not anymore, because in this updated version of the work, Dr. Shafiei used more than 40 different brain maps pertaining to neurotransmitter receptors and transporters, but also different layers, cell types, and then metabolism, different axes of cortical expansion, as well as different quantifications of local microstructure. So now we're looking at a whole lot of different functional characterizations and a whole lot of different microstructural and anatomical characterizations, one against the other, and trying to find common patterns that explain why they are where they are. Now, first, it is good to replicate one's own work, and this is indeed what Dr. Shafiei did. We had PC1 of those dynamical features, again, explaining a significant proportion of the variance and being associated, to perhaps nobody's surprise, with different spectral properties, because, of course, this is a well-known way of characterizing MEG activity in terms of spectral properties, and those local dynamical features correctly recapitulate what we know about the spectral properties of the MEG signal. So far, so good. And here comes the next step. This large-scale characterization, data-driven, using partial least squares analysis to map massive dynamical features phenotyping onto microstructural phenotyping. And lo and behold, we do find a significant principal component, and that associates different properties, again, primarily pertaining to the autocorrelation, but not just the autocorrelation. This is just a low-level representation of that large-scale characterization. And we find that many properties of microarchitecture are actually significantly correlated with that principal dimension that recapitulates the dynamical features obtained from MEG, including patterns for microstructure, but also the gene and gene pre-C1 and the T1-T2 ratio, which is a representation of the anatomical hierarchy. So again, anatomically, hierarchically organized. That's how we can represent this large-scale dynamics in a low-scale representation in space. So altogether, we find that if we perform a fully data-driven characterization of local dynamics in the brain, there is high correspondence between hemodynamic and neurophysiological time series properties. We also find that there is a dominant axis of these neurophysiological dynamics, and that is co-localized with multiple features of brain microarchitecture. So once again, there is an embedding of these dynamics in the anatomy of the brain. And last, we can ask, how do these properties that we know support brain function and cognition change when we are perturbing brain function and cognition, for example, via anesthesia? So this is the setup. We take different individuals who are either awake or undergoing anesthesia, and in both cases, we systematically characterize the local dynamics of every brain region across thousands of time series, nearly 7,000 of them, and then we ask, what is the difference between those? Which features are changing the most, and where are those changes? And when we look at that, we're not limited to looking at the human brain, because although we have now been spanning different modalities, functional MRI and MEG, we've been so far constrained to the human brain, but we don't have to, because these techniques, this HTSA, highly comparative time series analysis, can be applied to data from any species, can be applied to data from any modality. And fortunately, anesthesia can also be applied to any species, because there is no known species that cannot be anesthetized, at least no known animal species, I should clarify. And the same anesthetics that work on you and me will also work on a macaque, a mouse, perhaps even a C. elegans. And so that's exactly what we're looking at. And the question becomes, across different anesthetics, across different species, does anesthesia change the way the local neural units behave over time? And look that this, we're using functional MRI for all of the mammalian species, but we're using calcium imaging at a similar temporal resolution for the C. elegans. So that's single neuron resolution. We're not looking just at hemodynamics. We're not looking just at patches of cortex. We're looking at local neural units, whatever they might be in that species. And we find that indeed, across different species and different anesthetics, there are very similar patterns of change in those dynamical features. The features are arranged in the same way for every species, and we see that some features are significantly reduced across every species, some features are significantly increased across every species. And one other aspect that you might notice is that most of this seems to be almost agnostic to where the change is happening. The main direction of change is by far in terms of features, not in terms of space. But we were asking, is this really true? Is this just our eyes deceiving us? Can we find some consistent spatial patterns? And it happens that for the human, macaque, and mouse, we do have some shared local annotations, namely gene expression. So we had about 80 brain-related genes that are shared and available for the human, macaque, and mouse. And so just as before, we perform a purely data-driven characterization of all of those genes in every species across every region against the corresponding patterns of anesthetic-induced changes in local dynamics. And we find that there is one significant component that goes beyond what is explained by spatial autocorrelation in each of those three species, and this is what it looks like in the brain. There is a significant anterior-posterior pattern of how anesthesia changes the distribution and the quality of local dynamics, regardless of a species and regardless of the anesthetic that is used. And if we look at what those features are, we find again the same culprits as we had seen before. Autocorrelation and the predictability of the signal. It looks like the signal is becoming less predictable over time. Space and time are becoming decoupled under anesthesia. And if we look at the genetic part, we see that we have, for example, parvalbumin, a marker of inhibitor interneuron, which many of those anesthetics act upon. But we also have markers, for example, of hypocretin, which is one of the clock regulators of the sleep-wake cycles. Makes sense in the context of this being anesthesia data. So altogether, we can use this large-scale data-driven approach to characterize across species, across regions, and across anesthetics, and across modalities as well, how anesthesia reshapes the dynamical landscape of the brain. And we can obtain data-driven phenotypes for that. And we can relate it to local annotations that are the same across every species to obtain, hopefully, mechanistic principles of how that is operating in the brain. And of course, once again, there is a lot more to be had than this, and now you have plenty of options to find out more. You can look at the preprint, which is also available through the QR code over there. If you want a talk that is dedicated entirely to this last part, then you can come to my talk on Saturday after clap night, but around midday, so hopefully you'll be back in a more cognitive state by then. And you can also come to my poster on Friday and Saturday. So yours is the choice for how to interact with this. But let us not forget that this is not my work. What I've been talking about is Dr. Golia Shafiei's work, and here are her acknowledgements, because for her, as for all of us, it takes a village to do this kind of work. So please join me in acknowledging Dr. Golia Shafiei's work.